Our next presenter is Ashley Hanas, who is a toxicologist with the Michigan Department of Environment, Great Lakes, and Energy.
Good afternoon, everyone. My name is Ashley. I'm an aquatic toxicologist at the Michigan Department of Environment, Great Lakes, and Energy. And today I'll be presenting the results of our preliminary bifenthrin investigation. So out of the 1,000 chemicals that we have to think about on a daily basis, what is bifenthrin? So just in case that someone doesn't know, it is a broad spectrum perthroid pesticide. So perthroid pesticides are the synthetic counterparts to perthrin pesticides, which are derived from the chrysanthemum flower. Bifenthrin is used in a variety of commercial, residential, and agricultural settings. It's pretty hydrophobic, so it doesn't like water very much. It first partitions to sediments in aquatic systems. Accordingly, it also has a low water solubility. And if it does partition to sediments, it will persist for a relatively long time in the environment, with literature suggesting half-lives of 6 to 16 months. So in target and non-target organisms, bifenthrin's mode of action is to disrupt the voltage-gated sodium channels in nerve cells, which leads to prolonged nerve excitation and eventually can result in death. Bifenthrin is considered to be highly toxic to aquatic life in the water column, with low EPA aquatic life values for both vertebrates and invertebrates. It has also shown toxicity in aquatic sediments, with Weston and al. Reporting an LC50 of 520 micrograms per kilogram of total organic carbon for Hyla azteca, which also happens to be the screening value that is used by California and Washington. And also a more recent paper from USGS reported changes to insect emergence, community composition, and trophic cascade. The characteristics of the trophic cascade were essentially decreased graper abundance due to bifenthrin concentrations, which reduced the grazing pressure on periphyton, and then resulting in a higher chlorophyll A concentration. So as you can see based on the table here, that these effects were reported at concentrations previously believed to be safe for aquatic life. So one of the driving forces behind this project was essentially to better define the absence and presence of bifenthrin in Michigan water bodies. So that just leads us to our objective, was just to get preliminary data. Is there bifenthrin in Michigan? And if there is, what are our concentrations? So now you might be wondering, especially after seeing those really low water quality values, why are we looking at sediment? Well, as I mentioned before, bifenthrin is hydrophobic, so it doesn't remain in the water column for too long. So, and we also selected sediment because bifenthrin has been known to accumulate and persist in the sediment as well, so it gave us more flexibility in our sampling and also gave us an increased likelihood of detecting it, which was our goal for this project. As mentioned on the previous slide, there have also been several studies noting toxicity for benthic organisms, and they are exposed to bifenthrin via contact with sediment and pore water, and also ingestion of sediment-associated particles. So after selecting our media, we had to determine where we wanted to sample, which is a bit of a needle in a haystack. There are areas in Michigan that are extremely ag heavy, so where the potential impacts of pesticide haven't been well defined. So we wanted to use this opportunity to get more data, which led us to select 6 ag-heavy sub-watersheds within the Pine, Shiawassee, and Raisin Huck 8 watersheds. And we also had the opportunity to investigate a more urban developed sub-watershed within the Ottawa-Stoney Huck 8 watershed. And this was attractive to us as bifenthrin is a product that is readily available over the counter at your average home improvement store. And it's also a product that is frequently used by pest control applicators. In our specific sampling locations, we essentially honed in on those by seeing what areas were accessible to via road crossings, and then also potential of sediment deposition in those areas. So once we arrived to a site, we would collect the sediments from depositional zones, and we collect with a ponar or a stainless steel spoon, and then we put it all in this big stainless steel bowl, and then we essentially made mud soup and aliquoted it into requisite sample jars. And then, as per laboratory protocols, we kept the samples on ice after collection. So once we got our results back, the first thing we did after QA/QCing was standardize the bifenthrin concentrations in the sediment by applying the TOC, or total organic carbon correction. And this was just to make the results comparable amongst the different water bodies and also the other studies that have examined bifenthrin in sediment. So once our TOC corrections were applied, our concentrations were compared to the screening values developed by Washington and California. And we also used the results of the USGS study due to the ecological relevance of the endpoints. So another aspect we kind of wanted to look at, just to maybe streamline future investigations, is to see if there's any obvious connection between bifenthrin and land use. So we pulled the 2025 cropland data layer and generated a 100-meter buffer around our sampling locations. And we examined essentially percent agriculture, percent natural, and percent developed as well. And these relationships were assessed with linear models. And now kind of going into our results. So essentially the next couple slides are going to be the maps of our sampling locations, just to get a better idea of like the distribution that we did find. And to orient you, the legend, green is non-detect, obviously. Yellow is when we found bifenthrin that was below the screening values we established. The orange indicates it's within that USGS study range, so 197 to 233. And the red was above the USGS study range, so higher than 233. And this red asterisk is essentially concentrations that were higher than 520. So in the Pine and Shiawassee, the water bodies we sampled ended up actually being pretty close together, so they're all kind of lumped in here. So these two, Bush Creek and Rook Drain, are water bodies in the Pine, and then we had Beaver Creek and Shad Creek and Bad River in the Shiawassee. And then the Raisin watershed, we did Bear Swamp Creek, and we also did Little River Raisin, Camp Drain, and Brennett Drain. For our urban watershed, the Ottawa Stony, we did 3 water bodies, and those were Pitts Creek, French Creek, and Plum Creek. So just to kind of summarize all the information that was in those maps, we had 62 sites sampled total. However, unfortunately, we did have matrix interference present in 14 samples, so we had to get rid of them. Not get rid of them, but we omitted them from the maps and the statistics. 6 of our samples actually exceeded 520, which is the Washington, 520 micrograms per kilogram of TOC, which is exceeding California and Washington screening values. 8 of our samples fell beyond the 233 micrograms per kilogram of total organic carbon, which was the USGS screening line. And then within that range, 197 to 233, we only had 1 sample fall within that sampling range. But still kind of fun results, I guess. And 55% of our samples did have detectable concentrations of bifenthrin. To go in a little bit more depth, our highest concentrations were found in Little River Raisin, so that was in the Raisin watershed. You can see it's pretty eggy, and so this was essentially an egg ditch that we sampled from. And then we also had Plum Creek, which is our more urban and developed watershed. And then Bear Swamp Creek, which is back in the Raisin. Again, we have an egg ditch that we pulled this concentration from. However, proportionally, the water body with the highest frequency of bifenthrin concentrations was in fact Plum Creek, so from that more urban developed Ottawa Stony watershed, with 4 to 4 samples having detectable levels of bifenthrin. And looking at it from a Huck 8 watershed perspective, we found that the Raisin had 54% detection frequency of bifenthrin, and then the Pine was 25%, the Shiawassee was 42%, And then our urban watershed, Ottawa Stony, had 100% detections. And as for a land use analysis, our model indicated that if you have a higher percentage of medium intensity development, you tend to have higher bifenthrin concentrations. However, again, this needs to be interpreted with caution. Our sample sizes weren't as robust as we wanted them due to the matrix interference. So our model was pretty weak, and the P-line was borderline. However, this finding is supported by the literature, where other studies have found bifenthrin being the main driver of toxicity in urban sediment toxicity tests, bifenthrin being the most frequently detected pesticide in urban sediment, and also bifenthrin being present in urban and stormwater runoffs at concentrations known to result in toxicity to aquatic organisms. So we found it. Yay. Now what? So absence and presence is only half the battle. Now we need to kind of determine the widespread concentrations and try to figure out potential impacts to organisms. So obviously we want to kind of better refine our land use models, so we want to investigate additional urban watersheds. We would like to see if we can obviously capture it in the water column. So we would have to go immediately after a rain event to get that pesticide pulse before it partitions to the sediment. Or if we're lucky, maybe we'll find an auto sampler that can do that for us. And then we also want to kind of better tease apart bioavailability and bioaccessibility of bifenthrin in these aquatic systems. So we'd like to collect some water quality measurements and also some additional sediment endpoints. And then to complement all that, we would like to do macroinvertebrate surveys to assess potential biological impacts of sediment bifenthrin concentrations. And this one's maybe a bit farther down the road because this can be costly, but we'd also like to do sediment toxicity tests. In Michigan, the way a water body can be listed as impaired due to sediment contamination is through sediment toxicity tests. So we'd like to work towards that as well. So the main takeaway, bifenthrin is occurring at concentrations within sediment that could cause altered insect emergence, changes to community composition, and also trophic cascade. We do need more data to clarify the relationship between bifenthrin use and land use in Michigan, and additional endpoints are necessary to better understand bioaccessibility and bioavailability. So this is like the first step of what we hope is many. So obviously my colleagues, I couldn't have done this project without them, deeply appreciative of them. They went to stinky egg ditches on really hot days, dug out stinky soil, and none of them complained even once. So I think I was probably complaining the most. So I couldn't be more grateful to these guys. That's a quick reference slide, and happy to take any questions that you may have. Thank you.
We do have time for some questions. Oh, we have questions up front.
Yeah, since you had so many interference problems with kind of, I'm assuming the whole data set, wouldn't you also want to sample some of the farmland and some other things instead of just doing the urban sampling?
Yes, so a lot of our matrix interference was actually in the urban samples. So, which is another reason why we'd like to expand the urban data set, but we do want to do more ag investigations as well in different parts of Michigan to try to, again, get that data set really much more robust so we can make more connections with land use and bifenthrin.
Hi. So I know that you mentioned that you are lacking some information regarding bioaccessibility and bioavailability. I'm curious, if you do a little bit more research, do you think you might have enough to potentially perform a risk assessment to see if this is at risk for potential exposed populations?
Yes, absolutely. Some of the literature does have, already does have some pretty good ties to bioaccessibility and bioavailability of bifenthrin, but the goal is, like you said, the risk assessment. We want to apply it specifically to Michigan waterways and kind of better define the risk in Michigan. So if we do that investigation, I think we'll have a better understanding of potential impacts of bifenthrin, and obviously, at least bifenthrin maybe being one of them, because these waterways, there's never just one chemical, right? So we can at least try to maybe tease apart bifenthrin's role in potential impacted invertebrate communities.
I have a question from online. Sarah Nedrick asked, is the urban presence due to tick treatments or prevention? Thoughts on prevalence in urban?
So that's a great question. So I think, so with the urban, there's a lot of, there's a lot of pesticide applicators there that will do treatments on buildings, and so ticks could definitely be one of them, but it could also just be lawn treatment for, or just, I know some areas will do mosquito control as well. So it could be from that. It could also be from just people having access to bifenthrin and using it a little, maybe probably a little bit irresponsibly, and yeah, using a lot of that as well. But it's a great question. I'm not exactly sure on why it could be so prevalent, but those are my assumptions and theories so far based on what I've learned.